This final video is putting everything that we've done about protein synthesis all together on one piece of paper. So my advice is just get a blank piece of paper and come along my drawing journey with me. So to begin with, protein synthesis begins in the nucleus. So what I'm going to do is I'm going to draw myself a bit of a nucleus over here. Now it's got nuclear pores as well. I'm going to label it nucleus down here. If you want to, you can label a nuclear pore, and that's going to make a bit more sense when we get there. So then on the outside of the nucleus, this is the cytosol. So we are working completely within a cell, and these are the two main parts of that cell. So within the nucleus, we start with a piece of DNA, and I'm going to draw it already kind of unzipped a little bit, like this. Okay, so this in here is DNA. You can see it's got a lot of bases in it. And the very first thing that happens in transcription is that the DNA unzips. Okay, I'm doing all the DNA in one color like this. So the very first thing is that number one, the DNA unzips. Okay, what's going to happen next is RNA polymerase is going to come along and bind itself to the template strand. So number two, RNA polymerase binds to template strand. Now that RNA polymerase is going to move down the molecule here, and eventually what's going to come out the back of it is number three. It's going to, this is going to be a bit tricky for me to try and draw this. Just do that. Too many lines. There we go, getting there. All right, so as the RNA polymerase draws down here, you can see it's creating this complementary sequence of free nucleotides. In the nucleus, you've got a whole bunch of free RNA nucleotides. So these are free RNA nucleotides. Yeah, and this here that's being produced is a pre-mRNA molecule. Going back to what number three is, so the free nucleotides join to make pre-mRNA. Okay, and that's pretty much these pink stages here. That's what we write down for transcription. That's all happening around the site of the DNA in the nucleus here. Now, I know I haven't given myself very much space for my nucleus, so what I'm actually going to do is I'm going to make my nucleus a bit bigger. I might make it like this. Okay, so my nucleus is going to come out like this. And I'm still going to have my nuclear pore, just to give myself a little bit more space. Sorry. So now we've got this pre-mRNA molecule, and we've got to do a couple of things to it. Number one is I'm going to add, and this is where I like to draw a little bit of a picture. This is my methyl cap. Add the methyl cap, get it? It's like a cap for your head. We're also going to add number two. Sorry, guys. Number two is going to be add a poly A tail. And number three is going to be splicing. And that splicing process is going to remove introns and join exons. And what you're going to end up with is a little tinier molecule of mature mRNA, like this one here. This is now mature mRNA, and it's ready to leave the nucleus. So all of this stage, so you had stage number 1 over here was transcription. Stage number 2 over here in green is RNA processing. Okay, with 1, 2, 3. And then the final stage we're going to go through is going to be the stage of translation. I'm just figuring out what color to make it. And that final stage is going to happen when the mature mRNA comes over and joins with a ribosome, or a ribosome comes to join with the mature mRNA. Now, the whole reason that we have to make this mRNA that I didn't really talk about before was the fact that when DNA is a big molecule, you can see it's a big molecule here on the left. It's a big pink molecule, and it's not going to fit out one of these nuclear pores. The mature mRNA, on the other hand, single-stranded, nice and short, just one gene, easily leaves the nucleus. So this is kind of one of the main reasons that we do transcription, to create this mature mRNA. The other reason is you don't want to ruin your resource of DNA. Now, you don't want to send it out to the ribosome, possibly get it tangled up and it gets destroyed. Then you've lost your reference. So think of DNA like your centralized massive library or big server. You don't want to damage it. So you can copy it as many times as you like. We can go through this process of transcription and RNA processing and use all of that mRNA out in our cell. But it doesn't make any difference to the DNA. The DNA will stay there in the middle of the cell and be your reference point, and you can always create more mRNA. So what we do in our final stage is we end up, the mature mRNA moves over to the ribosome, or a ribosome goes through. So this here is my mature mRNA. And it's read in 3s, it's read in codons by an by a tRNA molecule, with an amino acid on the back of it. And so what we get here is we get number 1, we get binding to the ribosome. Number 2, we're reading in 3s. Number 3, the tRNA brings a specific amino acid, and that's based on the genetic code. And then number 4, a polypeptide chain of many amino acids is formed. And this final purple stage, that is translation. So you can see here, I'm just going to change this color here to make it a bit prettier. You can see here that each one of these stages can be represented on one diagram. We've got the inside of the cell, where we've got inside the nucleus, then we've got outside in the cytosol, and all of these create these polypeptide chains. Now, we are going to talk about what happens to a polypeptide chain after it's been created, but this is an overall schematic diagram showing protein synthesis.